Plasmolysis. Have a container and it contains highly concentrated solution It can be a sugar solution or a salt solution. Place a cell in this solution. The cell has cell sap. Cell sap is nothing but cell solution. This cell sap or cell solution also must be having some concentration. See that the concentration outside the cell is more than the concentration of the cell sap. Naturally, what happens? If it is higher concentration, its water potential will be less, and here because it is low concentrated, the water potential is more. So, from high water potential level, that is from lower concentrated solution, water goes out, means the water from the cell comes out. We call it osmosis. As a result of osmosis, what happens? When the cell loses water, naturally the protoplasm shrinks. When it shrinks, say here is the cell and here is the cell wall. And this is plasma membrane abutting the cell wall. And here is protoplasm. And this vacuole of the cell having cell sap. Now it is placed in a hyper concentrated solution. The water from the cell sap goes out. The protoplasm shrinks. Then what happens to the protoplasm? The plasma membrane is a part of the protoplasm, and that protoplasm separates from this is plasma membrane. This is cell wall. This is a plasma membrane. Gradually, the vacuole inside the cell shrinks. The protoplasm remains attached to the cell wall at certain points. What are those points? There are adjacent cells. The protoplasm is connected with the protoplasm of the adjacent cells by what are called plasmodesmata. So, at the place of plasmodesmata, the plasma membrane may not detach immediately, but at the remaining parts it gets detached. And when there is a further loss of water from the cell, the protoplasm totally gets detached and it forms a lump inside the cell. And this space between the cell wall and the plasma membrane is filled by the solution present inside the beaker, that is hypertonic solution. This contraction of the protoplasm and separation from the cell wall, this process is called plasmolysis. So this cell is said to be a plasmolyzed cell. If this plasmolyzed cell is placed in water, that is hypotonic solution, then again the water can move in and the cell gradually expands and occupies its normal position. That is called deplasmolysis. So when cells are placed in hyperconcentrated solution, cells get plasmolyzed, and if the plasmolyzed cells are placed in hypotonic solution, they are deplasmolyzed. In living body of a plant, never plasmolysis takes place. Plasmolysis is only an external feature. That is, experimentally we can prove it and we can make it, the cells plasmolyzed.